Welcome to Outbreak Brief, the podcast that takes you inside the world's most dangerous infections.
And today we are looking at a pathogen that honestly feels like science fiction.
Yeah, we're diving into a threat that doesn't just infect the human body, but essentially colonizes the very rooms and, the life-saving medical tools meant to heal you. It's a silent, multidrug-resistant fungus that seemed to just emerge out of nowhere all at once.
Right, all over the globe.
So imagine you're an investigator, right? But instead of dusting for fingerprints at a crime scene, you are combing through this massive global database. It's called the Sentry Antifungal Surveillance Program.
Which is a huge repository. I mean, scientists recently went back and analyzed something like 20,788 fungal samples collected from patients worldwide between 1997 and 2016.
Yeah, they were looking for the genetic footprint of a very specific killer. But when they looked at the data before 2009, they found a ghost. Out of those tens of thousands of samples, there were what, maybe 4 retrospective cases?
Basically zero. It essentially did not exist in human medicine.
And then suddenly, it was just everywhere.
It is one of the most perplexing biological mysteries of our lifetime. The genomic data we're looking at shows a threat that forced the entire scientific community to rethink how a global outbreak actually begins.
Right, so our mission in this deep dive is to unpack a massive stack of epidemiological reports, CDC tracking data, and phylogenetic studies. We need to figure out what this thing is, how it engineered itself to beat our best drugs, and, well, what it means for the future of medicine.
So the pathogen we're tracking through these sources is called Candida auris, often abbreviated just as C. auris. Biologically, it's an Ascomycetes yeast.
Which sounds like a mouthful.
It does, yeah. But it simply means it's a type of fungus that typically forms its spores in tiny microscopic sacs. Out in nature, these types of fungi usually just help break down organic matter.
And the name auris comes from the Latin word for ear, right? Because the very first time it was officially isolated and identified was in the ear canal of a 70-year-old Japanese patient back in 2009. Now, I need to pause on that because reading through the initial research, I found myself getting genuinely confused. When I hear the word yeast or candida, I think of incredibly common, harmless things.
Right, like a standard yeast infection.
Yeah, or just the microflora that naturally lives in our gut. But the World Health Organization recently elevated C. auris to a critical priority pathogen. How does a standard yeast make that kind of leap?
Well, the confusion is totally justified because most Candida species are what we call commensal organisms. They share our biological dining table without causing any harm, living peacefully in our gastrointestinal tract or on our mucosal surface.
Like the lining of the mouth.
Exactly. But C. auris fundamentally rejects that behavior. It does not want to colonize your gut. It has this bizarre, terrifying affinity for human skin and for abiotic, non-living surfaces.
So it behaves much less like a standard natural yeast.
And almost entirely like a highly transmissible, hospital-acquired superbug.
Wow. Which brings us back to that Sentry database mystery. Because the genomic analysis, looking at the single nucleotide polymorphisms, the organism's DNA fingerprints, it revealed something that feels impossible.
Yeah, it didn't start with one patient zero who caught it and hopped on an international flight.
Right. This fungus emerged almost simultaneously on 3 completely different continents as totally distinct genetic families.
And standard epidemiological models just simply do not account for that. You do not see genetically distinct clades of a novel human pathogen spontaneously appearing in completely different hemispheres at the exact same time.
It's crazy.
It is. Yet the phylogenetic tracking identifies 5 major independent origins. Clade emerged in South Asia, primarily India and Pakistan. Clade 3 appeared in East Asia, starting in Japan.
Then clade 3rd in South Africa.
Right. Clade 4th in South America, specifically Venezuela. And more recently, Clade 5 was discovered in Iran.
I kept thinking of it like a coordinated global bank heist, where the robbers strike Tokyo, Caracas, and Pretoria at the exact same millisecond. If they aren't communicating, what is the external signal telling them all to strike?
Right. And according to the environmental research in our stack, the leading hypothesis is actually a changing global climate.
Which is fascinating.
It's the most biologically sound explanation we have right now. I mean, for millennia, the ancestors of C. auris were almost certainly harmless environmental fungi, just quietly living their lives in wetlands or high salinity aquatic environments.
But as global ambient temperatures steadily rose, those specific wetland environments began to change.
Exactly. They got hotter, and as water evaporated, they got saltier.
So these fungi were subjected to immense selective pressure. It's like an evolutionary training montage. They either adapt to the heat and the salt, or they die.
And they adapted beautifully. They upregulated certain osmotic stress proteins to handle the salt and heat shock proteins to survive the rising temperatures. And by doing so, they inadvertently solved the biggest obstacle to infecting a human being.
The mammalian thermal barrier.
Right. Our resting body temperature of 98.6 degrees Fahrenheit naturally roasts the vast majority of environmental fungi. They simply cannot survive inside us.
But because C. auris spent decades adapting to extreme environmental heat in those wetlands, our body temperature was suddenly no longer a barrier. It was just like a comfortable room temperature.
Exactly.
But there's a missing link here. How does a heat-adapted wetland fungus cross over into a human hospital in London or Chicago?
Well, the jump likely involved intermediate animal hosts. Migratory birds frequent those exact warming wetlands and could easily carry these newly adapted fungal strains across entire continents.
And didn't they recently find it in a dog, too?
Yes. C. auris was recently isolated from the oral cavity of a dog in Kansas. That strongly points towards zoonotic transmission animals shedding it into rural human populations. From there, it colonized people silently, traveling on their skin until those people eventually interacted with the healthcare system.
And once it gets into that healthcare system, the environmental adaptations it made in the wetlands turn it into a total nightmare. Our sources note it's incredibly halotolerant, meaning it loves salt, and thermotolerant, loving heat.
Right.
So if I'm thinking about the human body, our sweat glands are essentially prime real estate for this thing.
That's exactly it. The environment of the human axilla, the armpit, and the groin perfectly mimic the harsh, salty, warm conditions it evolved to conquer in the wetlands.
Gross, but fascinating.
Yeah, and a colonized patient will shed millions of these yeast cells directly from their skin onto their hospital gown, their bed sheets, and the surrounding medical equipment.
Which is where its ability to survive on abiotic surfaces comes into play. The reports show it persisting for weeks, even months, on plastics and stainless steel. Ventilators, patient bed rails, thermometers, intravenous catheters. How is it surviving on dry plastic for a month?
It creates microscopic fortresses. C. auris is highly adept at forming biofilms. It essentially secretes this dense, glue-like extracellular matrix around itself, anchoring it to the plastic or steel and protecting it from drying out.
So healthcare workers touch these surfaces, pick up the biofilm on their hands, and carry it to the next room.
Exactly. But it's important to clarify who is actually on the receiving end of that transmission.
Right. The data emphasizes that a healthy person walking down the street or even visiting a hospital is generally not at risk. This is an opportunistic pathogen.
Right. It waits for a compromised host. It preys almost exclusively on the sickest of the sick. We're looking at intensive care unit patients, individuals with severe underlying conditions like diabetes, chronic kidney disease, or HIV.
Heavily immunocompromised people.
Exactly. And the absolute highest risk comes when those patients require invasive medical devices.
Because things like mechanical ventilators, central venous catheters, and feeding tubes literally bypass the body's natural defensive barriers. They provide a physical highway for the fungus to travel straight from the skin into the bloodstream.
And looking at the timeline of the data, the tragedy of the COVID-19 pandemic becomes glaringly obvious. The pandemic practically built a red carpet for C. auris.
Yeah, the biological overlap was devastating. During the height of the pandemic, you had overwhelmed hospitals and a massive influx of critically ill patients on mechanical ventilation.
But the medical treatments themselves played a role too. To save people from severe COVID-19 inflammation, doctors heavily utilized steroids, which suppress the immune system's white blood cells.
And they also prescribed massive amounts of broad-spectrum antibiotics to prevent secondary bacterial infections.
Right. But antibiotics only kill bacteria. They do absolutely nothing to a fungus. So by wiping out all the normal, healthy bacteria in a patient's body, doctors unintentionally eliminated the biological competition.
So a biological vacuum was created.
Exactly. And C. auris rapidly filled it. We saw massive, deadly spikes in C. auris co-infections within COVID-19 ICUs across Italy, the United States, and India.
So what does that actual infection look like? The clinical descriptions outline a terrifying progression. As long as it just stays on the skin, a person is colonized but completely asymptomatic. You'd never know it was there.
Right, but once it takes that highway down a catheter and breaches the bloodstream, it triggers fungemia.
And fungemia is the presence of the fungus multiplying in the blood, right?
Yes, and from there, it acts like a systemic poison. It can seed into major organs, causing endocarditis and infection of the heart valves, or intra-abdominal infections. It can even cross the blood-brain barrier and cause meningitis.
And the trickiest part for the doctors seems to be its disguise. The clinical signs of this invasion are high fever, severe chills, and a dramatic drop in blood pressure.
Which, if you're an ICU doctor, looks exactly like standard bacterial sepsis.
Yeah, the mimicry is where we lose precious time. A doctor sees a crashing patient, assumes bacterial sepsis, and immediately pushes broad-spectrum antibacterial drugs.
And days pass while the patient continues to deteriorate, and the medical team doesn't realize they're fighting a fungus until those standard treatments completely fail.
Because of this delayed recognition, the mortality rate for invasive C. auris infections is staggering. The studies in our stack show it ranging anywhere from 30% to 72% in adults.
Up to 72%.
That's horrifying.
Yeah.
And a massive driver of that mortality rate is that even when doctors finally realize it is a fungus, the drugs they reach for simply bounce right off it.
Right. Our pharmacological arsenal is failing. The clinical breakpoints from the CDC show that roughly 90% of all isolates are resistant to fluconazole. About 30% are resistant to amphotericin B, and 3% to 5% are resistant to echinocandins.
Could you break down what those three drug classes are actually supposed to do?
Sure. So the azoles, like fluconazole, are designed to stop the fungus from synthesizing ergosterol, which is basically fungal cholesterol necessary for their cell membranes.
Okay, so that targets the membrane. What about the second one?
Amphotericin B is a much harsher drug. It physically binds to the membrane and tears microscopic holes in it, causing the cell to leak and die.
And the echinocandins.
Those are usually our absolute last line of defense. They inhibit the fungus from building its rigid outer cell wall.
So we have drugs that stop membrane production, drugs that tear holes, and drugs that break the walls. How is CRS dodging all three of these mechanisms?
Through incredibly sophisticated genetic mutations. To beat the IUs, CRS mutates a gene called ERG11, which literally alters the shape of the cellular target so the drug just can't bind to it.
Wow.
Even wilder, it heavily upregulates efflux pumps. These are transmembrane proteins that act like tiny, highly efficient cellular vacuums, pumping the drug out of the fungal cell the second it enters.
It just spits the drug back out.
Exactly. And to beat the echinocandins, mutations in the FKS1 gene change the structural makeup of the cell wall, rendering the drug useless.
And reading through the latest updates, the most chilling detail is that we're now seeing pan-resistant strains. Strains that have mutated to block all three classes at once.
Yeah, pan-resistance is a nightmare scenario.
If the drugs literally do not work, what is the protocol when a patient is crashing?
It forces clinicians into experimental, highly desperate territory. Typically, an echinocandin monotherapy drugs like caspofungin is the first-line empirical choice. If the strain is resistant to that, they pivot to liposomal amphotericin B.
But if they're facing a pan-resistant strain,
Then doctors have to try synergy. They combine different classes of failing antifungals, hoping that the combined toxic stress might somehow overwhelm the fungus's defenses.
which begs the obvious question, if treating it is this much of a nightmare, why aren't we focusing on immunity? I mean, we rolled out multiple COVID-19 vaccines in under a year. Why does the research pipeline for a C. Echinocandins vaccine look so empty?
The challenge comes down to a fundamental difference in cellular biology. Viruses and bacteria are vastly different from human cells, but fungi are eukaryotes.
Meaning they have a true nucleus, right?
Yes, on a structural and metabolic level, a fungal cell is incredibly similar to a human cell.
So it's like trying to invent a poison that only kills the weeds in your lawn.
Exactly, but leaves the grass perfectly green, except the weeds and the grass share 90% of their DNA.
There's a brutal biological hurdle.
It is. Designing a vaccine or a systemic drug that attacks a eukaryotic fungal cell without causing massive toxic collateral damage to the host's own cells is immensely difficult.
And natural immunity offers very little protection, right?
Almost none. Even if a patient survives the blood infection and is discharged, it takes an average of 8 months for their immune system to naturally clear the silent colonization from their skin.
8 months.
Yeah, and inside a healthcare facility, they rarely clear it at all, remaining silent reservoirs. So instead of vaccines, the pharmacological pipeline is hyper-focused on developing completely novel, first-in-class antifungal drugs like risafungin and abrexifungin to try and bypass those mutated afflux pumps.
So if we can't vaccinate
Yeah.
and we struggle to treat, our only true weapon is infection control. We have to keep it out of the bloodstream entirely.
Right, but stopping the spread requires knowing it's in the room. And the diagnostic data in our sources outlines a massive structural failure in our hospitals.
Standard diagnostic machines literally cannot see this fungus accurately.
Yeah, early detection has been critically compromised by our own technology. Most standard biochemical testing machines used in hospitals, like the early versions of the Vitex 2 system, rely on metabolic profiling.
And when they analyze C. auris,
They routinely misidentify it as completely benign common yeasts, like Candida hemolonii.
so an ICU team runs a test, the machine tells them they have a harmless yeast, they don't bother isolating the patient, and the superbug quietly spreads to every bed in the ward.
Precisely why advanced diagnostics are so urgent. Hospitals need DNA-based sequencing methods, or mal-de-Topf mass spectrometry, which uses lasers to analyze the protein footprint of the pathogen.
But if a facility doesn't have that expensive machinery, what do?
They have to rely on specialized culture mediums, like Chromagar Candida Plus. When you swab a patient and grow the sample on this specific chemical agar, C. auris colonies react to the medium and turn a very distinct light blue color with a blue halo.
So it allows lab techs to visually spot the threat.
Exactly.
Okay, let's say they spot the blue halo. They know it's in the room. They move the patient. Now they have to clean the environment. The guidelines say that standard hospital surface cleaners fail completely against C. auris. Throwing like quaternary ammonium disinfectants at it is useless.
Right.
Why doesn't standard hospital cleaner kill a yeast?
Because standard hospital disinfectants are primarily designed to disrupt thin lipid membranes, the kind you find on many bacteria or enveloped viruses. But C. auris has a thick, complex, chitin-based cell wall, completely reinforced by that sticky biofilm matrix we discussed earlier. No, it can't. To destroy it, facilities are forced to use harsh oxidizers. They have to use high-concentration chlorine-based products, like chlorclean at 1,000 parts per million, or vaporized peracetic acid.
You essentially have to chemically burn it off the medical equipment.
Yeah, and that is incredibly labor-intensive. It requires strict transmission-based precautions, staff wearing heavy gowns and gloves for every interaction, and active surveillance where nurses are constantly swabbing the armpits and groins of new admissions just to screen for it.
And that level of protocol is exhausting for a healthcare system. Which brings us to the current surveillance data. We are losing ground.
We really are.
Looking at the 2024 CDC tracking numbers, there were 6,304 new clinical cases reported in the United States alone. To give you some perspective, back in 2016, there were exactly 48 cases.
That is an explosive, exponential leap.
And when you look at the geographical map of the U.S., the burden is heavily concentrated. California reported over 2,500 total clinical cases. New York had over 2,200, Illinois over 2,200, and Nevada over 1,700. But then you look at states directly neighboring them, and the map shows 0 cases. Is the fungus actually stopping at state lines, or is the map lying to us?
The map is showing us an illusion of infrastructure. There are certainly genuine, dense outbreak clusters in those hotspot states. California, New York, and Illinois have massive, highly interconnected healthcare networks.
Right, particularly long-term acute care hospitals, or LTCHs.
Exactly. Which act as central hubs where chronically ill patients are transferred back and forth, dragging the fungus with them. But the zeros on the map in other states do not mean the fungus isn't there.
It just means they aren't looking hard enough.
Or they usually just lack the advanced mal-de-Topf laboratory infrastructure to aggressively screen and identify it. And globally, the hidden burden is massive. Recent studies show it now accounts for 10% of all Candidemia blood infections in South Africa and up to 5% in Indian ICUs.
So we have a climate-adapted, multidrug-resistant fungus quietly spreading through the global healthcare system, hiding on ventilators, and dodging our best drugs. Based on the phylogenetic data, what is the scientific community's biggest fear for the future?
The most terrifying possibility lies in its genetics. Earlier, we mapped out the 5 distinct global clades. Fungi reproduce, and within these C. auris clades, there are different mating types, specifically known as MTLA and MTL-alpha.
Okay.
Currently, clades 1 and 4 possess one mating type, while clades 2 and 3 possess the other. Because these clades originated on different continents, they historically never interacted.
But because of global travel and interconnected healthcare systems, Strains from Clade 1 and Clade 3 could eventually end up in the exact same ICU in London or New York.
Yes, and if a highly azole-resistant strain from one clade and a highly echinocandin-resistant strain from another clade meet on a patient's skin, they have the biological hardware to sexually recombine.
Meaning they could swap genetic material.
Right, and give birth to an ultra-virulent, entirely pan-resistant hybrid superstrain that carries the most lethal mutations of both parents.
That is a staggering thought. It underlines exactly why the push for rapid point-of-care diagnostics is so critical. We need tools that work right at the patient's bedside, not just in an advanced lab, to catch these strains before they ever cross paths.
Absolutely. And it shows how desperately we need a way to safely decolonize a patient's skin without relying on toxic chemicals.
Those are the immediate tactical challenges. But if we pull back and look at the absolute biggest picture, the most sobering insight from all these sources is what Candida auris represents. It is a messenger.
It's the first multidrug-resistant fungal pathogen that we believe was directly birthed by a changing global climate.
It adapted to survive the heat, and in doing so, it learned to survive us.
Which leaves us with a deeply unsettling question. As global temperatures continue to rise and remote environments continue to shift and dry out, what other harmless, microscopic organisms are currently adapting in the wetlands right now, inadvertently engineering themselves to break the mammalian thermal barrier?
It's a reminder that the environment doesn't just change around us, it changes the things that live within us. Something to think about next time you read the CDC reports. Stay informed, stay prepared. This has been Outbreak Brief.